Okay, hi everyone, good afternoon. Thanks for joining us today in our workshop, Vieques Behind an Environmental Crisis. My name is Gabriela Terandino, and with my teammates Marily Ruiz Corte and Patricia Colomdia, we will be talking about a project in iGEM Room UPRM. Next slide, please. We are iGEM Room UPRM, the first and only team currently right now in Puerto Rico, from Puerto Rico and the Caribbean, composed of 19 undergrad students, 2 graduate advisors, and 2 PI advisors. Last cycle was our first year competing, and we won several medals and a nomination for Best Open Track. On the screen on the top, you can see the current team in one of our virtual meetings. And at the bottom, you can see the 2019 with the medals in Boston. Next slide. As a team, we do not only just work in our genetic prototype, we also are working currently in educating our Puerto Rican public what synthetic biology is by doing workshops and educational conferences about it just like this one. That's why this year we made the second SynBio 101 camp, this time completely virtually, impacting over 50 high school students from all across Puerto Rico, giving them the tools they need for working with synthetic biology. Our slogan, Synthesizing a Better World, as you can see on the screen, makes us always think of what we can do to help others via synthetic biology. Having this in mind, this year we decided to research about what problems were currently happening in Puerto Rico. Next slide. Vieques is a municipality island at east of Puerto Rico, and you can see it on the left of your screen. Despite being that little tiny island, just like Puerto Rico, Vieques is home to many different species, making it real biodiverse. Because of us, Puerto Rico being a U.S. territory, 2/3 parts of the island were used for U.S. Marine military practices. And because they were practicing, the bombs and the shooting could get caught in Vieques' homes. At the right on your screen, you can see a residual bomb that exploded in the past few days in Vieques. After 6 decades filled with constant bombing, the island protests were finally heard, and the U.S. Marine left the island. Although they physically left, they left behind hundreds of contaminants in the islands, harming Vieques' biodiversity, touristic areas, and Vieques people's health overall. Next slide. By us, iGEM Room UPRM, helping the U.S. Marine clean Vieques, we are going to be helping every aspect of it. This postcard was made by one of our teammates, Elimar, and she wanted to represent what Vieques meant for us, our team, and what we want to Vieques to look in the future. On the left, you can see how we see Vieques right now, really dark and just like ugly with all of these contaminants. And on the right, you can see how we see Vieques in the future using our project, just beautiful as it is. Now, my teammate Marieli is going to talk to you about the contaminants that can be found in this island.
Some heavy metals and organic compounds found in Vieques are mercury, RDX, PCBs, arsenic, lead, and uranium. They're usually components of military explosives and are very toxic to the human body and the biodiversity that surrounds us. And I want you to remember that these are just a few of the hundreds of contaminants found in the bodies of water and soil in Vieques, Puerto Rico. Some effects on the human body are cancer, asthma, cardiovascular problems, organ damage, and brain damage. And this has seen that the risk of cancer in Vieques is 27% higher than the rest of the main island. 34% of the residents have toxic levels of mercury in their bodies. 34% have mercury, 55 lead, and 65 arsenic. Many studies have shown throughout the years to track the heavy metals found in their bodies. High levels of heavy metals are found in the flora and fauna, which has caused a decrease on the agriculture and on the biodiversity of the island. In this slide, you can see some images of what Vieques have been dealing with. The top image is a pile of military parts and mortar shells that burns on the former naval training range. The left image is a bomb explosion, and the right one is a bomb that is located inside the ocean that has yet not detonated, which affects the biodiversity inside the ocean. Also, this kind of finding suggests that there is a huge decrease in agriculture since the soil is contaminated, it is not possible to eat fruits and vegetables grown inside of Vieques. Anones Lagoon. Anones Lagoon is one of the most contaminated lagoons in the world and in Vieques. It is known to be a toxic serum since it is directly connected to the Caribbean water, meaning that every ounce of contaminated water can go directly to the open ocean. In addition, there has been a 90% reduction in marine fishing in the last 10 years. And why is this a problem? Well, Viequenses' primary food is fish, and their economy depends deeply from fishing. And as you can see, this is a problem that has been affecting every aspect of Vieques, including the health, the biodiversity, and the economy. Therefore, iGEMeetParis project will be focusing on the contaminants of RDX and mercury found in this lagoon. Now I'll leave you with my teammate, Patricia.
Hello, everyone. I'm going to be talking about a little bit about how the environmental cleanup is currently being done in Vieques and some of the problems they have encountered during this process and how we can use synthetic biology to help. Right now, the agency that oversees the cleanup is the U.S. Environmental Protection Agency. 4,000 acres of 10,000 acres of surface contaminants have been cleaned, but this is without including the bodies of water and underground surfaces, which also have contaminants. The agency has also declared that the cleanup will be complete approximately by 2031 and 2032, which is going to take about 11 to 12 years from now. And this is just a tentative date. It could take even longer. Some of the techniques being used right now to clean these contaminants are practice of open air burning and open air denutation. The problem with these techniques is that they take too much time. Open air burning can cause harm to the wildlife in these areas, and open air denutation can result in the uncontrolled release of toxic contaminants to the environment. And since it's a very small island, these toxic emissions can endanger public health of the residents in Vieques. So the people in Vieques not only have to deal with the contaminants left behind by the US Navy, but also new toxins that are being emitted by the cleanup. As shown in the picture, this is a movement that has been going on for a couple of years now to create awareness of the situation happening in Vieques and how the people are asking for safer cleaning method. Next slide, please. So we as a scientific community propose a different approach to help solve this environmental problem by using synthetic biology. Some strategies that can be performed by microorganisms to clean up man-made or natural disasters are bioremediation and biodegradation. Biodegradation is the process of using microorganisms such as bacteria or fungus to biodegrade, decompose, or transform contaminants, while bioremediation is the process by which microorganisms transform or alter the structure of chemicals found in the environment. Next slide, please. Here is an example of biodegradation and how you can integrate synthetic biology. This is a study done by the University of Washington, where they use bacterium Deinococcus radiodurans, which are one of the most radio-resistant bacteria. So they use this as a potential chassis and modify this organism to biodegrade toluene and mercury from nuclear waste areas. And I want to mention that this is actually a project that this university proposed to the US Environmental Protection Agency. And the agency even gave them grants to carry out this project. So this shows that they are open for new innovative ideas to help clean environmental disasters. Next slide. Some of the benefits of using these scientific approaches are that it can help decrease pollution in a more natural way. It can be, depending on the situation, a potential faster solution. Also, it can be adaptable since you can use bacteria that are natural in the area that you want to clean and modify them to biodegrade or bioremediate the contaminant of your choice without altering the surrounding ecosystem. This can also be applied to various environmental situations. For example, as Maria Lee mentioned, our team, iGEM RUM, is focused on biodegrading RDX and bioremediating mercury in Vieques. But if our prototype is successful, it can be used in other countries that have the same problem. This is our team spirit poster where our teammate Emily illustrated our beautiful island of Puerto Rico, where she also illustrated each one of our team members with their own unique personalities. On the right side, you can see that Vieques, and it zooms in on the biodiversity of the island. Although it's a very small island, as mentioned by Gabriela, it is enriched with immense biodiversity with our project, we aim to help create conscious of the situation going on in Vieques and propose innovative ideas to help diminish the contamination. This is all for today. I hope you guys enjoyed it and learned a little bit about the situation going on in Vieques and how we propose to use synthetic biology to solve it. Thank you for your attention, and if you have any questions.
Okay, thank you. Does anyone have any question for them?
If anyone does not have questions, we would like to ask questions to those who are here. Do you guys think it is an approach that is ethical or unethical?
Yes, hello. Can you see me?
Yes, yes.
Yeah, great. Thank you very much for this presentation. It was very nice to hear from you. To answer your question, I think it's like GMO, and it's a complicated question to know whether you can release a bacteria that is modified in nature. But I definitely think that within laboratory, it's an interesting work to do. And I think to release in nature, you need to have a very solid reflection and like experimentations around how it will impact the ecosystem. And as you mentioned, you need to know also how it will impact the flora, the fauna, water and soil. And yes, maybe also have something to control the population that you have released in nature. This is the first thing that come to my mind.
So we have our research director, Paula. She can tell you how we're gonna do it, kind of. So Paula, if you wanna say.
Yes, hello. Yeah, I agree with you that the purpose of the prototype is it should not affect the flora and fauna of the island, because our purpose is to actually help the flora and fauna by decontaminating the island and the waters and the soil. So yeah, I agree. And that's why like we're trying, like our plan is to do like a closed prototype that we're going to take samples and all that, so we don't release those modified organisms to the environment.
Another question we would like to make you guys is that what contaminants are found or are present in your city or country, and how this has been affecting the community and the biodiversity that is surrounding you guys.
Well, I know that there is a team in Paris that is studying the pollution in the Seine River, so the river that is crossing the Paris. So I guess this is a matter you can find in many rivers. I don't know what are the pollutants, but and yes, and recently you know that the Cathedral Notre Dame burnt, and it raised question about heavy metals being released in the city center. So for a while they closed all the schools in the city, and yes, so this is a problem, but which you cannot solve, I guess, by releasing bacteria in nature again. So it's more like chemical treatments and this kind of stuff. But I guess maybe in the future, if we have really targeted bacteria, this could work. I don't know if you have thought a little bit, how could this be implemented? Like, should they be restricted to a particular area, or should they be restricted by a kill switch they have within their metabolism? Or yes, have you guys thought a little bit on this aspect?
Yeah, but first of all, I would like to ask you, do you know what's causing the contamination in the sign?
No, actually, I don't.
Okay, it's okay.
Maybe
Well,
there's someone else
maybe
here.
I can like answer for
Yes,
that.
maybe
It's
Eve from Sorbonne or Chloe
yeah, I don't know
can
if you
answer better
hear
this
me.
question.
Do you hear me?
Yeah.
Okay, so well, yeah, the water is polluted and mostly with hormones, pesticides, and antibiotics. It's actually mostly from companies that have kind of rejected those hormones or pesticides, and it's also because well, the water is not enough recycled, and even after a treatment, you still can find hormones. For example, it can come from birth control. So it's mostly about companies, and because the treatment of water is not efficient enough.
All right. Well, yeah, it's really interesting to kind of like learn about like the contamination in other countries and other cities and like what causes them, because I think in general like they contaminate the waters, I think, all around the world. I think that's a pretty general thing. So yeah, it's pretty interesting. And what the other question, well, yeah, we're planning to put kill switch in the bacteria so they're not released into the water. And we also have like a backup plan in case like the kill switch doesn't work, to reduce the possibilities that the bacteria will be released into the water.
Maybe can you explain us your backup plans if it's like possible?
Yeah, of course. We were planning on using and implement UV light in our hardware, because that's a method um, that will eliminate the bacteria as the water is going out, as the clean water is going out of the device. So that will assure us that with the kill switch, and with that, it will assure us that the bacteria will not be released out into the water. And we're planning to do it with water first, like the decontamination of water, but we also would want to in the future adapt the prototype so we can do decontamination of soil, because in the island of Vieques, like the soil is also really heavily contaminated.
And for us, it's really important to know what problems other countries are facing, since our prototype, we're going to use it in Vieques, but it does has the possibility to be used in other places. So that's why we like to know what's happening in other countries or cities, so we can know that in the future maybe we can use this.
Okay, that's interesting. Have you heard also about in the Caribbean? I know that in the French islands in the Caribbean, they've been using for a long time a chemical product to grow plants, which is chlordecone. And today they're facing big problems with this pollution to the point that they cannot harvest anymore the lands, and it has come to the sea and pollute the fish and so on. So it's a little bit similar to your situation in Puerto Rico, but this time it's for agriculture and not military exercise. So yeah, it's the same kind of problematic in the Caribbean, but yeah, surely can be applied elsewhere as well. And I can see in the chat that iGEMeTaris has mentioned that in Greece it has actually been used to clear the sea already. That's interesting.
That's really cool, and thank you for the information. I didn't know that 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 agriculture in that case has caused contamination in the waters.
And what kind of approach will you give to the citizens, in our case will be the Viequenses, for them to collaborate with our project and do not see us as like getting control of them?
Well, this is interesting. And do you have, like, for example, a plan to do this? Because I think that this is really important, that the citizens accept the project and obviously not feel that it's an obligation for them to accept it.
We are doing a lot of educational programs to educate Puerto Ricans about what's happening, because especially the new citizens, the lower the younger people, they don't know that this was a huge problem in our country. So we're doing a lot of educational programs to educate the people about how this contaminant has been affecting throughout many years, and it's still going to affect us for many, many more, and why this project is very important and will be good for everyone.
I think, moreover, if people are facing a greater danger, they might be more open to new ideas, including GMOs, which is not the case. Like for if there is not another danger, people will like just are afraid of GMOs and would prefer things to remain as they are. But I think it's yeah, it's a question also of what is the greater risk, and finding a compromise to get a solution. So yeah.
And I think there's a lot of misinformation about GMOs also, that sometimes right, como que citizens decide to believe like what they want to believe, and I think there's a lot of misinformation about that also. Yeah, like it's not perfect, but it also does have positive sides to it. So and also like they choose to believe the media instead of looking for scientific papers and actually looking for the right information. So I feel like that's also what happens.
Yeah, maybe I'm thinking that the fact that there's only the media and the scientific information, having iGEM members that are able to make an intermediary between like scientists that are in the laboratory and that are like a little bit outside of this like common society, is a good way to spread new ideas and spread your point of view. I mean, at least facilitate the dialogue. I think that's I don't know if this is one of the purpose of iGEM founders. Maybe it is.
Yeah, definitely dialogue, I think, is very necessary.
And have you had discussions already about, like, with citizens? Did you have the opportunity to like connect with children or make classes or having this kind of human practice?
Well, right now, due to the pandemic and Vieques being a really vulnerable city because they don't have a hospital either, it kind of was a really responsible thing to do that we in Puerto Rico, the big island, we kept in here and we quarantine here. So right now we are doing all of it online. Right now we are trying to communicate with people, um, with uh, with teams there that have helped develop and have helped educate their public, so we can help and connect and have like a collaboration of how we can educate them so they can educate the Viequenses. We're kind of like in that process right now because of quarantine, but hopefully in the next year, the next cycle, they can go to Vieques and they can explain face to face, because we know it's different when we have it on a phone or even here in Zoom meetings. I mean, I think it's different when you have it like face to face and they know we are not lying to them, because that's something else that we need to understand and we need to take accountability for, because scientists before have said, oh, we are going to do this and do that and we are going to um, make your life better, but then they left and they never came back. So Viequenses are hurting from the contaminants, but Viequenses are hurting also because people have told them before, we are going to help you, and they haven't. So we have to rebuild that trust and educate them too about synthetic biology, because as my teammates had said, and I have stated before in the presentation, we are the first team from Puerto Rico and the Caribbean. So no one else really is talking about synthetic biology. Now, as a team, we are educating people so they can educate people and building this like a really big collaboration all over Puerto Rico so everyone can know uh, what synthetic biology is and what it's capable of. Yeah.
Yeah, and probably the fact that you're from Puerto Rico is facilitating the trust to be built. Like, it's not scientists from other part of the world that will come and leave after. And
I would like to add about what Gabriela said about the communication with Viequenses. Since the pandemic came early in the year, by the time we have decided the project, the pandemic was almost here. So that even though Vieques is part of Puerto Rico, they're a little island just beside. So for us to get there or they come to us, we have to go by boat. So since the pandemic, that transportation has only been for Viequenses. So we're technically not allowed to go to Vieques personally and physically. So that has been a really challenge. However, we have been doing, like Gabriela said, many projects to educate others and try to communicate via meetings and with other people that have already conducted some research inside of Vieques. And well, even though it has been a challenge, we hope that, like Gabriela said, that next year we can go.
Okay, and I have a question for you. Well, you feel really passionate about your project, so maybe you are planning to continue this project outside of the competition, or maybe it will be the next iGEM RUM team who will do it?
Hopefully, like right now we're doing the phase 1 and phase 2 project because of the pandemic. We don't have access to the lab. So for sure next year, then next iGEM year, we're gonna be working on the project. Hopefully something more like than just theory and papers. You know, like we can go to the labs and all that. But it would be amazing if we could actually take the idea out of the lab and actually implement it in Vieques, because the impact it will have, I feel like, is really positive.
And we definitely feel very passionate about this project because it's really close to our home, and we have seen many stories about what happening, testimonies about people that have lived there, had lived when the bombing exploded just beside their house. And I'm gonna send a link via the chat. It's a little video about 12 minutes. If you're interested, you can see it, and it explains perfectly what's happening, what happened, and what is still going on. So maybe in your free time you can watch it. I really like the video. It's not that long, but it's very, you can see how everyone is passionate about this.
Yes, of course. We'll go check it.
Okay, so I don't know if anybody has another question, or if you, iGEM room, have another question for us. But otherwise, well, we can like stop here if no one has another question.
Maybe you can share the video on the chat, in the chat, Marieli.
Hey, I'm looking for it. Give me a second.
Okay. Don't worry, we can wait a little bit. Otherwise, like, what would be the side effects of this bacteria? I think it's supposed to biodegrade mercury or heavy metals. Do you think that it can be like detrimental to degrade too much heavy metals, or that it can mutate in some way that could be dangerous for the environment? Do you have any perspective on this?
I feel like those are always gonna be like possible dangers about using modified bacteria to, especially to like using bacteria to clean the environment. But that's why we're doing the models and all that to try to lower those levels of danger. So yeah, I feel like we would do anything possible to try to like predict what's gonna happen. So like so those things don't happen, or if they happen, like at a lower level.
Yeah, okay, I see. And but
I
is
see
there
that
sorry?
that Natalia is talking, but we can't hear her.
Sorry.
Can you hear me now?
Yes, Natalia.
Okay. Oh, I was just saying that to add to what Paula is saying, like there is some things that we can predict or we can think are going to happen, but we're not gonna know until we actually do those tests, and we have that trial and error stage for our project. So I think for phase 2 of our project would be those new ways to discover what could we do to make those secondary effects less.
And also, since we're planning to put our prototype inside of a hardware, inside of closed space, then hopefully that means that it will not go out into the environment. And even though the water is passing through there, we can test it before it comes out So they're like, we're trying to add as many controls as we can to the project. We also have a kill switch inside the genetic circuits. So we're trying to do as much as control as possible, because we know that if one single thing goes wrong, it can have a lot of big effects. So that's also something we're implementing.
All right, thank you very much for everything, for this presentation and animating this workshop. Maybe Chloe.
Other invasive bacteria, while at the same time also competing with other conventional sunscreens, which we also know to cause other skin conditions like eczema. But maybe one of the most important reasons for this was actually in terms of coral health. So we know that sunscreens, and actually through our research, we did actually discover a surprising number of cross-polymers and copolymers of microplastics, which of course are quite regularly spread into the ocean in tons every year, in addition to conventional UV filters like oxybenzone and cinnamate, which again are believed to actually interfere with coral growth and further promote coral bleaching. So we wanted to try to find a solution both to that, and at the same time offer an improvement on skincare, while also maybe actually creating a new generation of cosmetics that could use bacteria as essentially a modular circuit.
Yes, so as Kaya said, our motivation for doing this workshop was because as we continue through this project, we recognized that the system that we were creating for a probiotic that could be spread on the skin could also be used for other skincare. So just to explain, so a probiotic is something that contains living microorganisms, usually bacteria, although sometimes you can have something like yeast. So this is beneficial in the cosmetics industry because the system can continually produce the active molecule rather than in terms of normal skincare in which you apply the mixture and that's it. So, so we want to look at the potential of synthetic biology to transform skincare industry, as well as the potential risks and downfalls, because throughout our project we have had a lot of conversations about biosafety and about potential risks that creating this type of system can have. So essentially we want to have a discussion about this to see what other people think, to see, and to kind of evaluate whether people think that this is a viable option for skincare, have a look at the different issues. So we're not going to be talking about our project in this. We've created 3 hypothetical scenarios for different skincare products. So this ranges from therapeutic to purely cosmetic, and then we'll have a discussion afterwards about the issues raised and the feasibility of this approach to skincare. So, Kais, do you think we should split into breakout rooms, or do you think we should just do it as a...
I think with the number of people we have, maybe just one room, one group together, we can discuss all the scenarios. Perhaps.
Yeah. Okay, so I guess we'll move on to our first scenario then. So, so this scenario is a topical probiotic on the face which will produce retinoids and hyaluronic acid. So this is an acne treatment. So you spread it onto the face and it should hopefully calm down the skin, prevent it from causing spots, causing blackheads, using E. coli. So essentially this is a therapeutic application. And then this scenario also contains a kill switch, so the bacteria will die after a certain amount of time and won't remain on the skin indefinitely. So we've put on a couple of questions, kind of things to consider about our bacteria. So the first one is, how would you ensure that this system is safe? So does anybody have any thoughts on that?
Well, for example, there is many labs working with L'Oreal, etc., that are working to test toxicity of some bacteria for the skin. But I don't know if they have some results yet. But one question that I have already is, for example, many cosmetic products have a conservative cream or some things to keep the product in store for many months. How would you manage to have some conservative product with bacteria?
Caius, do you want to?
So actually, my sound just broke out in that last few seconds. Could you just repeat the last part of your
Yes,
question?
because many cosmetic products have a conservative cream that to inhibit bacteria and contamination. And so your project has some bacteria and microorganisms on the skin. So you will have to develop a cream or product that can be stored for many months, while and in the same time keeping the bacteria active.
Yes, so well, that's certainly a challenge, and this is one thing that we have been researching, not least by actually looking at other probiotics that are on the market. Now, most of these do use a more common mixture of Staphylococcus epidermis, Firmicutes, the more common residential bacteria on the skin. But I imagine what we would use instead is very much more oil-based. So you'd use some of the more natural oils that are found in the skin. So in themselves, they wouldn't be antimicrobial, but it would allow it to be stored, maybe very much like Actimel, in a fridge. So maybe for example the expiration date might be 2 or 3 months, in which case you'd have to use the product in that time. It would also, of course, prevent contamination by refrigerating it. So you just slow down the metabolism enough in order to keep the bacteria alive until they're applied on the skin. Where again, it's an oily mixture, like an emulsification, that allows it to stay on the skin.
Could another approach be to use like sporulating bacteria that would germinate once you started using them?
That's a possibility, but we did fear that often the formation of endospores is usually quite a pathogenic trait among bacteria. So yes, we could use something like Clostridium. There are of course fears about possible toxins that are being released, or using a bacterium like that. Because they're more susceptible to pathogenicity.
That's actually what our team's doing. We're using Clostridium, and we're using a spore-forming bacterium, and it's non-toxic. It's sporogenes, which has been found to be in the human gut already. So that's how we've got around that kind of like sell-by date problem, where we were thinking of something like a drink containing like probiotics, but obviously this has the issue of like contamination or a sell-by date and storage issues. So that's how we got around that problem.
Did you encounter fears? There was an interesting research paper that I read actually about sporogenes suddenly acquiring pathogenicity islands, at least from other species of Clostridium. Did you encounter that?
Like horizontal gene transfer?
Yes.
Yeah, we've been looking into that. We actually got a team, like a mini team, working on that in particular. I think one of the ways that we're trying to think about it is to induce like some kind of kill switch or supply an alternative substrate where if this isn't available, then the bacteria will not be able to survive. Something to do with like how they're going to uptake different genes, and if some certain genes aren't present, then like it will just completely kill the bacterium.
And you're right, certain auxotrophic strains at least have been very successful in the lab. For example, making bacteria dependent on a very specific type of methylalanine has been quite successful. Which actually that actually goes on to the second question as part of the scenario of actually how do you include a function of the kill switch? In a sense, one of the challenges that, well, if you use a nutrient or specific kill switch, is a possibility of evolving beyond that kill switch, and actually finding a mechanism that ensures that all the bacteria at a certain point die, or at least the escape threshold for that bacteria is low enough as not to be concerning. And so, for example, our project was looking more into essentially almost a stochastic kill switch. So at a certain point, the system is stable in two states and can either be alive or dead, but it can never transition between being alive and dead. Seems like, yes.
So one of the things that we needed to consider during this was the evolution of the kill switch and also the evolution of the genes. So presumably with most kill switches, there's a selection against the function of the kill switch, because obviously the bacteria needs to survive. So yeah, so how could you get around this selection against the kill switch happening is kind of one of the things that you need to consider here.
Hello. Hello, Caius. Isabella.
Hello.
Yes, we were having a discussion right before with a team from Puerto Rico, and they insisted on the fact that you can never predict how it will happen, how you can make things safe, and their plan is to test it in a closed area. So in your case, I guess it would be reproduce the skin somehow, if that is possible, and test to see if it's safe, to see how the kill switch works. So to me, this is one important aspect, is that you cannot predict even with models, and that you would need to have experiments and like enough experiments. I don't know what this means, like how much.
Yeah. You need to actually look at the evolutionary stability of the kill switch within a closed system, don't you? But can you think of any, because for example, when we were thinking about kill switch function, we thought about incorporating multiple kill switches essentially. So if one kill switch fails, there's another one which should hopefully then work in order to kill it. So that's one way that you can, it's not about obviously knowing exactly what's going to happen, but it does lower the possibility of the bacteria actually out-evolving you essentially.
Maybe also I'm thinking to another idea. Is it possible to have several bacteria that regulate each other? So you can say that one bacteria is the kill switch of the other.
So you have multiple strains within the probiotic?
Yes.
That's a really interesting idea. So how would that work essentially?
I guess it would work like one bacteria depends on the other to get the nutrient or something like this. I mean, that's one idea. And
so then if one bacteria disappears, the other one will disappear as well?
Yes. So this is, I guess, a way to have the multiplication of kill switch, because you can implement in one bacteria and in the other, so you have even more way to kill the bacteria.
Maybe another approach would be like something like a paracrine signaling process, where essentially you have one strain of bacteria, but they're likely to express a certain toxin at different times. But when one bacterium expresses a toxin, this leads to a positive feedback and causes other bacteria in the vicinity to also produce the toxin, which then causes others. So in a sense, you cause the single population to undergo a fade based on just a few of those bacteria in the starting population.
Because in effect, it's its own ecosystem, isn't it, really? And ecosystems all depend on each other. They're not just, it's like a big tree where you can't just chop out one. I mean, like if you chop out one, it could affect the rest of the population. Yes. But I think it's so complex because there's so many factors in an ecosystem, especially in the microbiome, where we don't even really know what's happening. It's so difficult to predict. I think there's one way that this looks to be quite interesting. I think Amsterdam are doing it. They're trying to couple growth of the bacteria with the product, so that synthesis of the product is required for the bacteria's growth. So it's almost like that is the positive feedback that the bacteria will try to evolve to continue living, to continue producing this product, rather than going the other way and trying to survive the antibiotic or whatever thing you're trying to use to kill it.
Yeah, being coupled about the growth and not about killing it. And for what you said about the microbiome, I think that indeed, it's an ecosystem and you cannot add just any more bacteria that you can, but maybe design some bacteria of the existing ecosystem. For example, if you have a bacteria that is a Staphylococcus aureus and Staphylococcus epidermidis, and you want to design one of them, you will have also to design the other bacteria that are present inside the microbiome. And maybe, yes, as we've been saying, add a kill switch or something to link the two bacteria in their growth, so their population dynamics are linked. This would be interesting to me.
Yeah, I think that's the way forward, to rather than just like copy pasting loads of kill switches in and you end up at some point with like 100 kill switches. I think that there must be some way where you have to link them and couple them to depend on each other to really control it.
I have seen a couple of systems which involve signaling between bacteria. So the bacteria essentially are programmed to only survive or only reproduce, I think it was, if they can sense that there are other bacteria around them of the same type. So this means that, say, for example, our sunscreen, it's probably going to be used when people go into the ocean. So say that it washes out into the ocean, they can no longer sense any bacteria of their own type around them, so they don't reproduce and eventually die, which I thought was an interesting system, kind of along those lines.
In a sense, this is exactly how quorum sensing would work in many mammalian cell types. Like an epithelial cell cannot really survive if it's isolated. It really does need its neighbors for its own survival. Maybe another approach in that sense is actually the converse, trying to maintain the survival of the bacteria. We know that it's incredibly difficult for a non-native bacterium to inoculate the skin. So what approaches can we actually think of to actually allow the bacterium that's maybe foreign, either it's a different species or it's not adapted to living on the skin, to actually survive on the skin without being killed by the other bacteria, like bacteriocins that are released by native bacteria?
Right, I think we should probably move on to the second scenario. So this scenario is kind of similar. So it's, again, a topical probiotic containing a therapeutic with a therapeutic application. So this one will produce a weak steroid such as clobetasone butyrate, which treats conditions like eczema, rosacea. So in this one's a little different in that in this scenario, there is no kill switch. We just expect that the bacteria will be outcompeted by the native skin biome because it's less adapted to the skin. So obviously, it's a little different, and it's not as the biosafety there is also, there's also some interesting questions there. So I guess, does anybody have any comments on it?
I feel like a kill switch would almost have to be, simply from like a health and safety point of view, almost has to be included, because if you don't, you're just kind of running the risk of seeing what happens with the other bacteria and just letting it happen rather than you having any control over it.
Yeah. Do you think, in terms of the regulation, do you think that any country would ever allow you to create a product without some sort of kill switch in there?
Probably North Korea.
Yeah.
Can't see it
Probably
happening.
not somewhere like Britain or Europe, though, I suspect. So, so essentially, how can you think of any other ways you can make it more robust apart from adding a kill switch in? So maybe considering horizontal gene transfer.
I have team members working on this, not me in particular, but they're working on trying to control horizontal gene transfer.
So what
approaches are they taking?
Sorry?
So how are they trying to think about horizontal gene transfer? What steps are they taking?
That's a question for my teammates.
Yeah, because it's also something we've been considering as well. So one of the ways that we looked at was using non-conjugative plasmids. So these are plasmids that don't essentially contain the genes that can actually allow them to travel between bacteria. But obviously other types of gene transfer you can't control as easily. So stuff like transformation, the bacteria will just, you can control the bacteria from accepting new genes via transformation. You can't control other bacteria from acquiring genes from your modified bacteria.
Or via bacteriophages, which can also be a problem. I mean, another approach, therefore, in a sense, with part of this problem as opposed to using a kill switch, is essentially making your strain particularly sensitive. So depending on how long you want the product to be present, if you already know that it's likely to be outcompeted, and you make the strain, therefore, more likely to become ill, so essentially include a certain mechanism that prevents it from reproducing after a certain point, or you make its carbon cycle less efficient. Can you imagine that maybe being used instead of a kill switch to control your skin bacterium?
I think that's an idea, but then you have the downside of how it affects your product, like your metabolite production then, and how much an effect your treatment will have if you basically are making it weaker.
Although in terms of this one, steroids, if you apply them for too long or use too much, essentially they can have some, they cause skin thinning, they can cause some other effects within the body. So it's possible with this one you wouldn't actually want too much being produced. So another thing is, how would you control dosage with something like this?
That's a pretty good question. For us in particular, dosage isn't too much of an issue because we've got such a wide range of like before it starts to get anywhere near a dangerous level, it's such a high number that for us dosage isn't such a problem. Especially, I guess it depends if you're trying to go for a short-term treatment or a long-term treatment. You might want it to be more stable over long term and to really control and have an accurate dosage size, whereas for short term you could almost run the risk of it slightly overshooting or not having too much of an effect.
So I mean, would something like a feedback loop work, do you think? So it can sense how much of the steroid is actually on the skin already and then produces it if there's not enough?
That sounds like a good idea.
Would you want to control that? In a sense, you almost have three mechanisms of controlling that. The two most important being you either control in your genetic circuits the level produced, and then you have some form of repressor system. So either you repress it at the gene level, or you repress transcription, or you repress translation, or you have a certain process that actually prevents proliferation of your bacteria. So either way, you're controlling the amounts of your dosage, but one, you control the gene circuit, the other, you actually control how many bacteria are producing the product.
Yeah, that's true actually. Yeah, because it's not just the population. You can control within a certain bacteria how much to produce.
Yeah.
So how about modeling? So which parts of this system do you think it would be important to model? Which parts would be difficult to model as well?
Just before this, you can see that in the chat, someone spoke.
I can't actually.
Oh yes, maybe if you put an ingredient that doesn't allow your bacteria to produce a big amount of the steroids. Yes, I mean it may be difficult if, for example, your bacterium is to be present in the bloodstream. For example, it's going to attack a certain target tissue, maybe a tumor. It's a bacterium that you've designed to target tumors. It may be difficult to control concentration of a master chemical, for example, because it will just diffuse across the bloodstream. But that's a good idea, however.
Does anybody have any comments on kind of how to model it or anything? So for example, what we've been trying to model has been the interaction between our bacteria and the native skin bacteria, which has been challenging, obviously, because the skin bacteria does vary a lot between different people.
maybe it's very different from the reality, but you could use a prey and predator model. Like if you achieve to have the idea of the stability of your bacteria um, and the native bacteria, you can just use factors to use a prey-predator model.
Yeah. And that was, you're right, there was a model that was made quite successfully for that, by actually looking at phage therapy, designing phages to target a bacteria. You use exactly that sort of almost vodka Volterra approach to the matter. What steps do you think would be the hardest to model if you were to go about modeling the system?
I think it's much more difficult with bacteria because there is not a real interaction of prey and predator between two different bacteria, because it's only an idea of place and of what we can call food for them. So it's just about the possibility to live for it.
Yes. And in a sense, maybe an interesting model would be something like almost a competitive exclusion, where you look at the growth of two bacterial populations, and you assume that there's an interaction between them. And you therefore, of course, you'd experimentally determine those interaction factors. You'd almost create, somebody set up a matrix of terms for that. And then you'd see how those populations change over time. So, I mean, in terms of, in the grand picture of stability, how serious would you actually consider something like horizontal gene transfer compared to some of the other problems we might have surrounding, for example, just long-term maintenance on the skin, or actually toxicity of the surface? So how big a problem do you think horizontal gene transfer?
Maybe from the law perspective, it will be very difficult to produce something like this, because if you change the microbiome for a long time with this gene transfer, it will be likely that pharmaceuticals will be not very happy about this, to change the microbiomes for a very long time and not be able to reverse.
Yeah, I think that's very true. From a regulatory point of view, that I think it's just like a total no-no.
And by the way, since we talk about viruses and bacteriophage, do you think it will be possible to construct a kill switch with a bacteriophage, like something that some bacteria that have integrated bacteriophage, like prophage, and for a system like if you have too many steroid, it will activate the lytic phase, and so the bacteria will be killed.
Yeah, so that's such an excellent idea. Essentially, you can almost couple that with almost like a Cree-Lock effect. So you actually use the natural OMP proteins found to regulate whether the phage goes into a lytic or lysogenic life cycle, and that you can almost use that as like we were discussing before, the frequency dependence. So once the bacteria population gets too high, or as if the steroids reach too high a level, this is activated, and it causes a dynamic instability in the population. It causes the entire population to be killed due to the formation of more phages. So that's a very good way to control this.
But the question is this not leading to even more transformation? Like you start by designing the bacteria, then you design the bacteriophage, and then you will have to design even more like bacteria. So I'm putting in the place of a normal citizen. Like you can be afraid that this doesn't stop, and we get to something very dangerous in the end.
I mean, usually bacteriophages are highly specific. So if you're using maybe a very specific strain of bacterium, you might also use the culture the same highly specific bacteriophage. In which case, maybe your product is there isn't as much risk of horizontal gene transfer via transduction between different species of bacteria. But it might be difficult to sell that in the EU, for example.
Oh yes.
Sorry.
No, I just wanted to react about this. You've talked about this before. To apply this kind of thing in the EU, like UK or in the West, I think it's definitely for cosmetic industry. It's complicated, like to sell this kind of things. But as we were talking before with the Puerto Ricans, they have pollution problem that can involve at some point the life of other people. So if in the balance you have life of someone and using GMOs or bacteria modified, I think this can be a way to implement this in the EU. I mean, I'm not sure, but this is that wouldn't be legitimate to me. And in that sense, I wonder how people should be kept away from other people, because you know the bacteria can be transmitted from one people to another. So in the prospect of a clinical treatment, how could like this bacteria medicine could be implemented? And yeah, do you think it will be possible here in Europe?
Yeah, I mean, that's a really good question that I'm not sure there's quite much of an answer to at the moment. It's something that we've been considering quite a lot, kind of because we've been looking at the regulations in the UK and they are very strict. So yeah, especially something that'll be able to spread, which is actually something that comes up in our final scenario, which I'll just move on to now. So this final scenario is it's a purely cosmetic probiotic. So it produces anti-aging molecules such as hyaluronic acid and coenzyme Q10. So with this scenario, instead of using a non-native chassis, we use Staphylococcus epidermis, and there is no kill switch. The aim of this product is for it to integrate permanently into the skin microbiome and then permanently produce these anti-aging molecules. So the big question with this one is, I guess, is it ethical to actually have a permanently integrated genetically modified bacterium on your skin?
To me, the short answer is no, it's not ethical. If there's is to be something like this, it must be under control and in a room that is sterilized, and the one should not be able to go out of this, like the patient. It raises the sort of question that raises like transhumanism, like being enhanced human. So to me, it's unfair for those who have it, and it can lead to like I don't know, a lot of craziness that is endangering many people. So,
and you mentioned before that sometimes these systems, they would be able to spread from person to person. So hopefully the ones we described before probably wouldn't be able to, but this one probably could. So, I mean, is there any way to prevent this apart from incorporating a kill switch?
Maybe I just imagine a bacteria that needs another cream. For example, it needs like an acidoamines or something like that. And the people who really wants to have staphylococcus on his skin have to put another cream on him. And that way, even if the bacteria just go through another person, you won't be able to have the amino acids, so it would die. And in that way, even if it's spread from one person to another. The bacteria won't be able to live and to spread again.
I mean, that's a really interesting idea, to have an amino acid that that's required that isn't provided anywhere else but on the skin of the intended user. One other thing about Staphylococcus epidermis, though, is that it's not a pathogen as such, it's opportunistic. So occasionally, most of the time, it won't cause disease, but occasionally it can, usually in immunocompromised patients. You find Staphylococcus epidermis causing certain infections. So yeah, is genetically editing a native skin chassis, do you think that's a good idea, or?
Hi, the thing is, I think the problem of the skin is like, if you just put it on your head if you want the face to be young and everything, it's probably fine. But if you put it on your hands and on your arms and you rub someone like a baby, or if you want even someone your age, even if you have the cream Alice talked about, you can also rub a part of the cream, and so the bacteria will stay a few hours maybe. So that will be a problem. And also, it's really like if you put your hands on handles, like it's the same problem as for COVID. If you have your hands everywhere and you don't wash them and you spread it everywhere, it's not as bad as COVID, but it's the same problem. Like you can spread it even if you don't want to. So the biocontainment will be a big problem, I think.
So yeah, can anybody else think of any other unintended consequences there might be from designing a system like this?
One consequences that will be if you have a new bacteria on your your skin, where you take a shower or anything like that, you will it will be put in the water, and next you can have some environmental effects with the anti-aging molecules, and it will be maybe next gen micropollutants in the water.
Let me ask: do you feel that the concern for essentially integration of a new bacterium, native bacterium, as part of the skin microbiome, but do you feel that this would be reduced if it did not actually have any genetic engineering in place? So, like the transplantation experiments that have been done with lactobacillus, replacing parts of the intestinal bacteria, would you have a problem if you were to use a bacterium that had not been genetically modified, but you were transferring it from one patient to another, for example, permanently, ideally?
Personally, I would not, but in this case, we are talking about a microorganism that is producing a new molecule. So you will have to study the effect of these molecules for some environmental issue, like fish or maybe plants too.
Yeah. So I think we've mentioned this before, but do you think this would ever actually be allowed? Do you think consumers would even want this?
I don't think in Europe it would be allowed. Like we're always strict on the GMOs and everything, so I don't think it would be allowed. And even more for something that's not like health, like a genetical condition. Like if it's just cosmetics, I don't think it would be. I think it would be frowned upon. Sorry.
I think there is definitely a market for it because people are happy. I think people would be up for that definitely if it's something that looks after their health rather than a treatment. It's kind of things that people take to stop themselves aging. I think it's quite similar to some of the nutrition stuff like probiotics, like Yakult. People, I think people are pretty happy to take stuff like that because it's something to do with their health, and I think people believe in that kind of thing.
Mm-hmm. So do you think a purely cosmetic GMO probiotic is less acceptable than a therapeutic one, essentially? Do you think one that only improves your appearance, not your health, is people will be less willing to use that.
I think it's true in the idea, like in the concept, but technically the border is thin between health and cosmetics. You can see that there's even a branch that is called dermocosmetics, which is making dermatology and cosmetic at the same time. You have a famous brand that is called Pierre Fabre doing this. So yes, the border can be thin, and you can argue that sometimes using GMO probiotics on skin can be part of a health concern. And yes, what I wanted to add is also that probiotics on skin have already been used on for health reasons, but not genetically modified.
Mm-hmm.
So I think if the genetically modified bacteria is kind of approved, it could definitely be used at least first in health, then maybe in cosmetics. I think it's a possibility.
Okay. I think we might be running out of time, so yeah, so essentially I think we've covered most of these questions, but so do you think that genetically modified probiotics could actually be part of the mainstream skincare industry, or do you think that it's going to stay in kind of a niche area essentially?
I think that GMOs will probably come in first in cosmetics because I think that's an optional thing, and it will then become more of a growing trend for people to take it rather than in health. I think if people feel they're forced to take it, or that doctors are trying to push it, maybe there's a kind of like an anti-trust your doctor kind of a thing. Whereas if you start with cosmetics and it's optional, and then maybe say you get like celebrity coming on board and famous actresses and actors taking that kind of stuff and celebrities, it will become more mainstream and people won't view it as badly as they do now.
Mm-hmm.
So do you imagine perhaps therefore in the future a form of celebrity endorsement for genetically modified organisms, as you guys mentioned for your soft pack of Malva?
I think so. And I think there's like a kind of like a biohacking trend coming in where people are trying to better themselves and trying to extend their lifespan or make themselves appear better from a more cosmetic point of view and for their own health, rather than to treat like a disease. I think it's a bit more controversial there. People are more against it because they feel they don't have a choice. I think that's probably one of the main things.
At the same time, if it's from doctors, you can have more trust in the product than if it's from like Donald Trump using it. It's a celebrity. You trust him. I don't know. And other celebrities. I would be more trustful if it was from the medical area. I would be more likely to use it, I guess. Maybe it depends on people also.
Mm-hmm.
It can be a trend movement.
Okay, so I think
maybe just one final query, therefore. So, and that's simply where would you place GMOs, at least in a cosmetic context, in 15 years' time?
Well, I think as some of you are already doing, I think in skincare, I think sun cream is an avenue. Same with like anti-aging products. I think that will probably become one of the bigger parts of cosmetics, anti-aging, by treating yourself with a therapeutic bacteria.
So yeah, thank you guys for coming. It's been really interesting, and it's been really interesting to hear kind of perspectives that we haven't considered before. And yes, this, if you have any questions, want to talk to us about anything, we've got our Instagram, Facebook, LinkedIn, email. So yeah.
Thank you.
Thank you very much. It was very interesting to ask some such questions. Very good idea. Thank you very much.
Yeah, thank you to everyone who participated in this workshop. Thank you